So yesterday morning, a viewer was kind enough to email me a new research study that was published just about a month ago. The title of the study is Impact of Feralization on Evolutionary Trajectories in the Genomes of Feral Cat Island Populations. And just below the title, we have the names of the involved researchers, respectively. Basically, the entire goal of the study was to figure out whether feralization can bring about genomic changes to domestic animals the way domestication can to wild animals. So basically everything I've covered in my previous videos on the mega cats in Australia, the cats on the Kerguelen Islands, and the wildcats of Madagascar and Crete. For the purposes of this study, the researchers sequenced the DNA of 4 feral cats on 2 very isolated islands that are Dirk Hartog Island of Australia and Kaho'owale in Hawaii. Domestic cats were introduced in these islands in the last 200 years and currently have very limited to no human inhabitants. Although Dirk Hartog Island is used for ecotourism, visitor numbers are very low, and a genetic study on the feral cat population showed no indication for introduction of cats to the island in the near past. So the cats have been surviving and thriving in the wilderness of both islands for the last 2 centuries with little or no human interaction in between. Where have we heard this before, I wonder? The study aims to compare the genomes of the feral cats on these 2 remote islands, since the lack of human presence made them ideal for observing feralization without domestic cat gene flow. The researchers wanted to understand, number 1, the origins and differentiation of these feral populations from their ancestors. Number 2, the molecular changes from feralization compared to domestic and wild cats. And number 3, the evolutionary forces driving these changes in both island populations. Vegetation on both islands is generally sparse, low and open, indicating similar environmental regimes. Despite these similarities, Dirk Hartog is 3 times larger than Kaho'owale. In addition to the Australian and Hawaiian feral cats, the researchers also studied the genes of 8 European wildcats, or Felis sylvestris, from Germany, as well as 11 standard house cats from North America, Europe, the Far East, and Middle East. And here on this chart, we have the Hawaiian ferals in orange, the Australian ferals from Dirk Hartog in red, domestic cats from the Far East, Middle East, Europe, and North America circled respectively. And at the far left, we have the European wildcats in light blue. With the cat samples, the study investigated population structure, genetic differentiation, genetic diversity, highly differentiated genes, and recombination rates. And here, recombination rate refers to how often genetic material is exchanged between different cats. This rate is important for understanding how genetic diversity within these cat populations can lead to various adaptations and evolutionary changes. Higher recombination rates result in more genetic diversity, which can help the population adapt better. Conversely, lower recombination rates mean less genetic diversity and increase the risk of inbreeding and potentially limit the ability for the animals to adapt to changing environments. I have linked the study below in the description for you all to read in depth on your own if you wish to understand the specific materials, methods, or analyses used in the study. Otherwise, I will be skipping straight ahead to the results and findings of this study. The feral and wildcat populations showed varying levels of inbreeding, with the Hawaiian, or rather I should say the Kaho'owale feral cats exhibiting recent inbreeding, and Australian, or rather I should say Dirk Hartog Island feral cats showing moderate levels due to occasional gene flow, likely from occasional human releases over the years. The wildcats have the highest inbreeding due to ancient bottleneck events, while domestic cats show the least inbreeding, likely due to the admixture of various cat breeds. European domestic cats particularly have the highest recombination rates, indicating greater genetic diversity, while wildcats have the lowest. The Dirk Hartog feral cats, and I'm only calling them that as opposed to the Australian feral cats because I do believe you go out to the Australian outback, the cats there are most definitely a very different animal than the creatures these researchers found on Dirk Hartog Island. Well, the Dirk Hartog Island cats had higher recombination rates than their Kaho'oale Hawaiian counterparts. The Dirk Hartog Island cats may benefit from different environmental factors or gene flow, suggesting a complex relationship between environment and genetic diversity. The genomes of the Dirk Hartog and Kaho'oale cats have diverged significantly from their domesticated ancestors, reflecting unique evolutionary paths influenced by their environments, notably genes related to nervous system development and reproduction are highly differentiated, indicating relaxed selection in these areas. The Dirk Hartog Australian feral cats showed more differentiation in musculoskeletal genes, likely due to their diverse prey compared to the limited prey options for the Kaho'oale Hawaiian ferals. Overall, while both populations shared similarities, their evolutionary responses highlighted the unique adaptations feralization entails beyond a simple reversion of domestication traits. In conclusion, this study showed that the feralization of these island feral cats serves as a compelling model for understanding the evolutionary processes that occur when mammal populations are released from human control. The findings proved that feralization in cats is a complex process, which leads them down a unique evolutionary path with unpredictable outcomes. This aligns with what I have discussed in my previous videos regarding the megacats in Australia's outback, Madagascar, Crete, Corsica, and the Kerguelen Islands, reinforcing the idea that their highly probable evolutionary trajectories are influenced by complex genetic mechanisms shaped by their environments. The researchers highlighted significant genomic changes associated with feralization, specifically that adaptive evolution was less influential than the relaxation of domesticated traits in these cats. To further differentiate the genetic changes linked to feralization, I certainly think more research studies like this are necessary. Maybe next time consider examining feral cats in other various environmental and demographic contexts. Maybe consider one of these alleged wallaby hunter feral cats thriving deep in the Australian outback, or maybe even including the ancestor of domestic cats, Felis libica, or the African wildcat in this study would also help identify distinct genes across wild, domestic, and feral populations, and would certainly provide a deeper insight into the continuum of domestication and feralization. Thank you all so much for tuning back into Eclectic Explorations. Please continue to like so that the YouTube algorithm continues to notice me, share, and if you haven't already, please subscribe. I'll see you all next time.